Imagine we found a cure for everything, a way to instantly erase a genetic disease from existence. No more cystic fibrosis, no more Huntington's disease. We have the technology, and it's as simple as using a pair of molecular scissors. This is the incredible promise of gene editing, a tool so powerful it could end human suffering as we know it. But here's the profound question that hangs over this miracle. What happens when we go beyond fixing and start upgrading? What happens when we can choose our children's eye color, make them smarter or physically stronger? Today, GenShift is stepping into the moral labyrinth of designer babies. We're not just talking about science, we're talking about ethics, fairness, and what it means to be human. Get ready to confront the most fundamental dilemma of our time. Hit that subscribe button now to join us in navigating tomorrow's mind and the technologies shaping our world. 1. The promise: curing disease, ending suffering. To understand the power we now wield, we have to look to nature's own microscopic battlegrounds. The technology at the heart of this revolution, known as CRISPR, wasn't invented in a lab. It was discovered. For billions of years, bacteria have been fighting a silent war against invading viruses. To survive, they developed a sophisticated immune system, a genetic memory bank. When a virus attacks, the bacterium captures a snippet of the invader's DNA and stores it within its own genome in a special archive called a CRISPR array. This snippet acts like a most wanted poster. The bacterium then produces a guide molecule, a strand of RNA, that's a perfect match for the viral DNA. This guide attaches to an enzyme called Cas9, which acts as a pair of molecular scissors. Together, they patrol the cell. If that same virus ever invades again, the guide RNA instantly recognizes it, directs the Cas9 scissors to the target, and slices the viral DNA, neutralizing the threat. Scientists, in a stroke of genius, realized this bacterial defense system could be reprogrammed. By creating a custom guide RNA, they could direct those molecular scissors to any specific sequence of DNA in any organism, including us. This transformed a natural defense mechanism into the most precise and powerful gene editing tool humanity has ever known. This isn't science fiction, this is happening right now. In late 2023, history was made when the FDA approved the first ever CRISPR-based therapy, a treatment called Casgevy. Its target, sickle cell disease, a debilitating hereditary blood disorder that causes excruciating pain and organ damage, disproportionately affecting Black communities. For patients like Victoria Gray, the first person treated in a clinical trial, the results have been nothing short of miraculous. Doctors remove a patient's own stem cells, use CRISPR to correct the single faulty gene responsible for the disease, and infuse the edited cells back into the body. These corrected cells then produce healthy hemoglobin, effectively offering a functional cure for a lifelong illness. The success extends to other conditions like beta thalassemia, freeing patients from the need for constant, painful blood transfusions. Beyond blood disorders, researchers have used a personalized CRISPR therapy to successfully treat an infant born with a rare and fatal metabolic disorder called CPS1 deficiency, correcting the faulty gene in his liver and saving his life. Early clinical trials are even showing promise in restoring sight to patients with a form of hereditary blindness. For these applications, what we call somatic gene editing, where the changes affect only the individual and are not passed on to future generations, there is a broad and powerful ethical consensus. When a tool can alleviate so much suffering and correct the cruel typos of our genetic code, the moral argument to use it is compelling. It represents the pinnacle of medicine, not just treating symptoms, but curing disease at its source. 2. The slippery slope, from fixing to upgrading. But the same precision that can delete a disease can also add a desired trait. This is where we step onto the slippery slope, a descent that doesn't begin with a dramatic leap, but with a series of seemingly reasonable steps. The journey from therapy to enhancement is a slow, seductive creep into a profound moral gray zone. It starts with prevention. If we can cure a genetic disease, why not prevent it from ever manifesting? It seems entirely ethical to edit an embryo to remove the gene for Huntington's disease or cystic fibrosis. But what about editing a gene that only confers a risk for a disease, like the APOE4 gene linked to Alzheimer's or the BRCA genes associated with breast cancer? Most would still agree this is a noble goal. The line begins to blur when we consider conditions classified as disabilities but not life-threatening. For example, some forms of hereditary deafness. Many would see this as a condition to be fixed, but within the deaf community, deafness is viewed as a unique culture and identity to be preserved, not erased. Using gene editing to eliminate deafness could be seen not as progress, but as an existential threat to that culture. So the question becomes unavoidable. Who gets to decide which traits are defects and which are simply differences? From here, the slope gets steeper. What is the difference between preventing below average height and enhancing height to be above average? What is the difference between correcting a cognitive disability and boosting intelligence to genius levels? Suddenly, we are no longer talking about disease. We are talking about enhancement, choosing a child's eye color, increasing their muscle mass for athletic ability, strengthening their memory, or even altering complex traits tied to mood and personality. The line between what is considered normal and what is a flaw to be corrected becomes dangerously subjective. Is being shy simply a personality trait or a social anxiety disorder to be edited away? Who draws that line and based on what values? This gradual slide is amplified by a powerful psychological bias called the anchoring bias. Our minds don't evaluate each new possibility in isolation. Instead, we latch onto the first piece of information we receive, the anchor, and judge everything else relative to it. In this debate, the anchor is the noble, life-saving goal of curing sick children. That emotional and ethical anchor is incredibly powerful. When we are anchored to that idea, the next step, preventing a predisposition to cancer, seems perfectly reasonable. And compared to that, correcting deafness seems like a smaller step. And compared to that, maybe making a child a little taller or a little smarter doesn't seem so radical after all. We are no longer judging the act of enhancement on its own profound merits. We are judging it against the unimpeachable good of curing a fatal disease. Each step makes the next one seem less controversial. And before we know it, we have slid from a place of clear ethical consensus to a place of profound moral ambiguity. This psychological pull, this gradual normalization of the extraordinary, is what makes the slippery slope so treacherous. It isn't a sudden fall, but a descent so gentle we might not realize we're on it until we've reached the bottom. 3. The genetic divide, a society of haves and have-nots. A cure exists, but it costs $2.2 million. That isn't a hypothetical price for a future technology. That is the real-world list price for Casgevy, the CRISPR therapy for sickle cell disease. A competing therapy, Lyfgenia, costs $3.1 million. While companies argue this price is justified by the lifetime savings of managing the disease, often between $4 to $6 million, the reality is a staggering upfront cost that our healthcare systems are not built to handle. This isn't a future problem. The economic barrier to genetic equality is already here, and it creates a terrifying paradox. A technology developed to alleviate suffering could become the most powerful engine of inequality humanity has ever created. If these therapeutic and eventually enhancement technologies are only accessible to the wealthy, we risk creating a future that looks less like a meritocracy and more like a biological caste system. The gap between the rich and the poor will no longer be about access to education or resources. It will be written into their very DNA. Imagine a world with two distinct classes of human. The naturals, subject to the whims of genetic chance, and the enhanced, a generation of children born with engineered advantages, stronger, healthier, smarter, their privileged starting position guaranteed before they are even born. Can a society where the affluent can purchase genetic superiority for their offspring ever be considered just? This division wouldn't just be imposed by cost, it would be driven by powerful social and psychological pressures. As enhancement options become available, parents would face an overwhelming paradox of choice. The sheer number of potential upgrades would create immense anxiety and a paralyzing fear of making the wrong decision, or worse, of choosing not to enhance and leaving their child at a natural disadvantage. This anxiety would be fertile ground for the bandwagon effect. Once a critical mass of wealthy families begins enhancing their children, the social pressure for others to keep up would become irresistible. It would trigger a genetic arms race, where parents feel compelled to give their children the same advantages as their peers, not for health, but for social and economic competitiveness. The fear of being left behind would become a more powerful motivator than any ethical reservation. This is the ultimate danger, a society that splits at the most fundamental level. We could see the emergence of a permanent, heritable class divide, where social mobility is not just limited by economics, but by biology. It would be an inequality that could never be overcome, passed down from one generation to the next, creating a genetic underclass and cementing a new, terrifying definition of what it means to be a have or a have-not. 4. Beyond our genes, redefining humanity. This brings us to the final and perhaps most important layer of this dilemma. Beyond the science and the sociology, this technology forces us to ask a fundamental question. What does it mean to be human? If we can rewrite our own source code, what makes us uniquely and intrinsically valuable? If we can edit out our flaws, what happens to the resilience we build by overcoming them? If we can eliminate struggle, what happens to the empathy we learn from shared vulnerability? If our genetic makeup becomes just another product to be designed and optimized, what part of us remains authentic? There is a profound value in the genetic lottery of life. The very imperfections, variations, and unexpected challenges that shape us are what forge our character. Our species' greatest strength has always been its diversity, the vast, messy, and beautiful spectrum of human traits. The drive for genetic enhancement is, at its core, a drive for homogeneity, a narrowing of what is considered acceptable or desirable. And that narrowing could ultimately make our species less resilient. Our perception of this entire dilemma is deeply influenced by a cognitive bias known as genetic essentialism. We are psychologically predisposed to see genes as a person's essence, a fixed, immutable blueprint that determines who they are. This bias makes the idea of improving genes dangerously seductive because it feels like we are improving a person's fundamental worth. It also explains the deep-seated fear of playing God, because we intuitively sense that tampering with DNA is tampering with something natural and sacred, the very core of a human being. But this is not a problem for scientists and philosophers to solve in isolation. This is an urgent social justice issue for our time. For Gen Z, a generation that champions diversity, inclusion, and authenticity, the stakes could not be higher. The principles you fight for in the social and political arena must now be applied to the biological one. The conversation about the ethics of gene editing is really a conversation about whether we will use our newfound power to enforce a narrow definition of perfection or to protect the beautiful, chaotic diversity that makes us human. The greatest challenge we face is not mastering the technology of our genome, but mastering the wisdom and humility to use it responsibly. The ultimate question is this: will we use this power to eliminate our differences or to finally, truly appreciate them? Conclusion. The power to rewrite our DNA is not merely a scientific question, it is a philosophical one, a question that forces us to define what we value most about being human. We now hold in our hands the ability to end unimaginable suffering and, at the same time, the potential to create unimaginable division. This technology is a mirror held up to our society, reflecting both our deepest compassion and our greatest fears. The future of gene editing is not yet written. We as a society still have the agency to guide it, with wisdom, foresight, and empathy. And the conversation starts now. It starts with you. What line should we never cross? Share your thoughts in the comments below. And if you believe these are the conversations we need to be having, subscribe to GenShift and join the community navigating tomorrow's mind.